Carbon dioxide is made as a waste product by cells, and blood helps to transport that carbon dioxide from the tissues to the lungs, where we can breathe it out. Now, to facilitate this, blood has three important mechanisms to move carbon dioxide around. First, a small amount of carbon dioxide is dissolved in the plasma, which is the liquid portion of blood. Now, to calculate the concentration of dissolved carbon dioxide, you can multiply the partial pressure of carbon dioxide, or PCO2, measured in millimeters of mercury, with the solubility of carbon dioxide. The solubility of carbon dioxide is the amount of carbon dioxide that can be dissolved in the blood. And it turns out that in 100 milliliters of blood, 0.07 milliliters of carbon dioxide is dissolved per millimeter of mercury of carbon dioxide. In venous blood, the equation becomes dissolved carbon dioxide equals the venous partial pressure of carbon dioxide, or PvCO2, in millimeters of mercury times 0.07 milliliters of carbon dioxide per millimeter of mercury, per 100 milliliters of blood. And if we plug in the partial pressure of carbon dioxide in the veins, which is about 45 millimeters of mercury, we get 3.15 milliliters of carbon dioxide in 100 milliliters of blood. This works out to be about 5% of the total carbon dioxide transported by the blood. Now, another 3%, or about 1.89 milliliters of carbon dioxide in 100 milliliters of blood, is transported a second way. Carbon dioxide binds directly to the terminal amino acids of each of the 4 globin chains in a hemoglobin protein. Hemoglobin is the most abundant protein in the red blood cells, and each hemoglobin can hold up to 4 molecules of carbon dioxide. When hemoglobin is bound to carbon dioxide, it's called carbaminohemoglobin. Now, carbaminohemoglobin has a slightly different shape than hemoglobin, and it has less affinity for oxygen, and this is called the Bohr effect. This leads to slightly more oxygen becoming unbound and getting dropped off in tissues full of carbon dioxide, and this causes a shift to the right in the oxygen-hemoglobin dissociation curve. But the majority of carbon dioxide, about 90%, or about 56.7 milliliters of carbon dioxide in 100 milliliters of blood, is transported a third way, which involves turning carbon dioxide into a bicarbonate ion, or HCO3-. To get there, carbon dioxide first undergoes a chemical reaction with water to form carbonic acid, or H2CO3. As a weak acid, carbonic acid easily dissociates into hydrogen ions and bicarbonate ions. And these reactions are reversible, and can happen in the opposite direction as well. And while this reaction can also happen in the plasma, it's sped up in the red blood cell by the enzyme carbonic anhydrase to produce a large amount of HCO3- and H+. As bicarbonate ion levels build up in the red blood cell, the bicarbonate ions start moving down their concentration gradient and flow into the plasma. The shift in charge this creates is balanced by facilitated diffusion, an ionic exchange between bicarbonate and chloride, called the chloride shift. So chloride comes into the red blood cell as bicarbonate goes into the plasma. And here bicarbonate plays an important role in maintaining physiologic hydrogen ion levels in the blood, which ultimately affects pH levels. It does this through Le Chatelier's principle. In other words, when the hydrogen ion concentration gets low, the equation moves to the right, producing more bicarbonate and hydrogen ions. Conversely, when there are lots of hydrogen ions around, the bicarbonate will bind to a hydrogen ion and form carbonic acid, which then slips into water and carbon dioxide. Ultimately, the concentration of hydrogen ions doesn't shift too quickly in either direction, and that means that the pH stays stable. Now, carbon dioxide waste ultimately needs to make its way from the tissues to the lungs, and be removed from the body through expiration. So in the lungs, release of carbon dioxide from the blood happens because of a decrease in partial pressure of carbon dioxide and an increase of partial pressure of oxygen compared to the tissues. This means that all of the processes working to pick up carbon dioxide in the tissues are reversed in the lungs. As a result of higher partial pressure of oxygen, more oxygen diffuses into the red blood cells and binds with hemoglobin at its heme group. Now, even though oxygen doesn't compete with carbon dioxide and hydrogen ions for binding sites, it does alter hemoglobin's shape just slightly, and this reduces hemoglobin's binding affinity for carbon dioxide and hydrogen ions. This is called the Haldane effect. And it means that as more oxygen binds to hemoglobin, more carbon dioxide and hydrogen ions become unbound. Now, when there's a rise in hydrogen ions, more bicarbonate is needed to bind with it. So bicarbonate re-enters the red blood cell from the plasma, once again by flowing down its concentration gradient. Except this time it's going into the red blood cell. With all this extra carbon dioxide building up, partial pressure of carbon dioxide increases and drives carbon dioxide diffusion down its partial pressure gradient from the red blood cell into the plasma. And from there it goes into the alveoli, where partial pressure of carbon dioxide is 40 millimeters of mercury. Partial pressure of carbon dioxide in the alveoli and blood equilibrate about 20 times faster than oxygen. And this is largely because carbon dioxide is about 23 times more soluble than oxygen. This is super important because even when there's a buildup of fluid in the lungs, like in pulmonary edema, and it's tough for oxygen to diffuse into the blood, carbon dioxide can still easily diffuse out of the blood. Alright, as a quick recap, there are 3 ways for carbon dioxide to move around in the blood: dissolved directly into the blood plasma, bound to hemoglobin as carbaminohemoglobin, and chemically modified and dissolved in the plasma as bicarbonate ions. These processes of carbon dioxide being picked up in the tissues are all reversible in the lungs, and this leads to carbon dioxide expiration. Oxygen content is the amount of oxygen in a certain volume of blood, typically 100 milliliters. Oxygen binding capacity is the maximum amount of oxygen that can be bound to hemoglobin, abbreviated as HB, which is the main protein found inside of red blood cells, which is the main component of blood. As it turns out, there are 2 major ways for oxygen to move around in the blood. The majority of oxygen is bound to hemoglobin inside red blood cells, and a small amount is dissolved directly in the blood plasma. So the oxygen content of blood is the sum of these 2. Oxygen content equals hemoglobin-bound oxygen plus dissolved oxygen. Now, if you just wanted to calculate dissolved oxygen, you do that by multiplying the partial pressure of oxygen, measured in millimeters of mercury, with the solubility of oxygen. And the solubility of oxygen is the amount of oxygen that can be dissolved in 100 milliliters of blood. It has a constant value of 0.003 milliliters of oxygen per millimeter of mercury per 100 milliliters of blood. So the equation becomes dissolved oxygen equals partial pressure of oxygen in millimeters of mercury times 0.003. If we plug in a physiologic arterial pressure of oxygen of 100 millimeters of mercury, we get 0.3 milliliters of oxygen in 100 milliliters of blood. Now, that's not enough oxygen to meet the metabolic demands of the body. So that's where hemoglobin comes in. The oxygen binding capacity of hemoglobin is the maximum amount of oxygen in milliliters that 1 gram of hemoglobin can bind, multiplied by the number of grams of hemoglobin in 100 milliliters of blood. Each hemoglobin molecule can carry up to 4 molecules of oxygen. That's 1 oxygen molecule for each of the 4 hemoglobin subunits. Now, because each red blood cell carries a few 100 million hemoglobin proteins, that means that each red blood cell carries over 1 billion oxygen molecules. So that means that for each gram of hemoglobin, there's 1.34 milliliters of oxygen carried around. Now, normally the concentration of hemoglobin in the blood is 15 grams per 100 milliliters. So multiplying that out, we get 20.1 milliliters of oxygen per 100 milliliters of blood. So the amount of oxygen in the blood bound to hemoglobin is actually quite a bit higher than the amount of oxygen dissolved in the blood, which was 0.3 milliliters of oxygen per 100 milliliters. But hemoglobin isn't always 100% saturated or bound by oxygen. In other words, a hemoglobin protein might have none bound and be 0% saturated. It might only have 1 oxygen bound and 3 open spots for more that haven't bound yet, meaning that particular molecule would be 25% saturated. It might have 2 filled spots and 2 open spots, or 50% saturated. 3 spots filled and 1 spot open, or 75% saturated. Or all spots filled with 100% saturation. And the amount of oxygen that's bound to hemoglobin is directly proportional to the amount of oxygen dissolved in the blood. For example, at a partial pressure of 25 millimeters of mercury, hemoglobin proteins might be a total of 50% saturated. And at a partial pressure of 100 millimeters of mercury, they might be at a total of around 98% saturated, which means that most are fully saturated. So going back to the original formula for oxygen content, and plugging in some numbers like a blood hemoglobin of 15 grams per 100 milliliters, and 98% for the percent saturation, we get 19.7 milliliters of oxygen from hemoglobin-bound oxygen, and 0.3 milliliters from dissolved oxygen, totaling 20 milliliters of oxygen per 100 milliliters of blood. So that's the oxygen content of the blood, but it doesn't tell us how much oxygen is actually delivered to the tissues. Oxygen delivery is the oxygen content multiplied by the cardiac output, which is the amount of blood that's being pumped out by the heart each minute. Cardiac output itself is calculated as the stroke volume, which is the volume of blood pumped per beat from the left ventricle of the heart, expressed as milliliters per heartbeat, multiplied by the heart rate in beats per minute. So if we assume a heart rate of 70 beats per minute and a stroke volume of about 70 milliliters per beat, we get 4,900 milliliters per minute as the cardiac output, or more commonly 4.9 liters per minute. And we said that there's 20 milliliters of oxygen per 100 milliliters of blood. So that's an oxygen delivery of 980 milliliters of oxygen per minute to the body. Alright, as a quick recap. The oxygen content of the blood is the amount of oxygen that's carried in the blood, which is dissolved oxygen and hemoglobin-bound oxygen. Oxygen content can then be multiplied by the cardiac output to determine the oxygen delivery, which is the amount of oxygen that reaches the tissues each minute. The oxygen-hemoglobin dissociation curve shows how the hemoglobin saturation with oxygen, or SO2, is related to the partial pressure of oxygen in the blood, or PO2. Hemoglobin is the main protein within red blood cells, and it's made of 4 globin subunits, each containing a heme group capable of binding 1 molecule of oxygen. So that means that each hemoglobin protein can bind 4 molecules of oxygen. But each hemoglobin isn't always 100% saturated or bound by oxygen. A hemoglobin molecule might have no oxygen bound and be 0% saturated, called deoxyhemoglobin, and it'll take on a tense state shape, or T state. Or it might have 1 oxygen bound and 3 spots open, meaning that particular protein will be 25% saturated, or 2 filled spots and 2 open spots, 50%. It might have 3 spots filled and 1 spot open, or 75%. Or all spots filled and 100% saturated. All of these states where oxygen is bound to hemoglobin are called oxyhemoglobin, changing to its relaxed state, or R state, with each oxygen molecule that binds. And since there are millions of hemoglobin molecules in a single red blood cell, and millions of red blood cells, the hemoglobin saturation of oxygen is the average saturation among all of these proteins. Now it turns out that hemoglobin absorbs different wavelengths of light as it gets more and more oxygenated. A technique called pulse oximetry uses this property of hemoglobin to figure out the average oxygen saturation across millions of hemoglobin proteins. The main factor that influences oxygen saturation is the partial pressure of oxygen in the blood, measured in millimeters of mercury. So, for example, at a partial pressure of 25 millimeters of mercury, hemoglobin proteins might be 50% saturated, called P50. And at a partial pressure of 100 millimeters of mercury, they might be 98% saturated, meaning most are fully saturated. And when these points are plotted, the curve takes on a sigmoidal shape. In practical terms, the sigmoidal shape means that hemoglobin has an increasing affinity for oxygen as the number of bound oxygen molecules goes up. So binding that fourth oxygen molecule is a lot easier than binding that first oxygen molecule. This is called positive cooperativity. Around 60 millimeters of mercury, the vast majority of the hemoglobin subunits have bound oxygen, so the curve starts to level off. That's why in arterial blood, where the partial pressure of oxygen is around 100 millimeters of mercury, hemoglobin gets fully saturated with oxygen. And also why in the venous capillaries of tissue, where the partial pressure of oxygen is about 40 millimeters of mercury, hemoglobin is only about 75% saturated with oxygen. In other words, about a quarter of the oxygen that's bound to the hemoglobin gets dropped off, or unloaded in the tissues. Now, there are a few factors that can cause hemoglobin's affinity for oxygen to change. For example, when carbon dioxide is produced during aerobic metabolism in the tissues, it dissolves into the blood plasma, increasing the partial pressure of carbon dioxide, and increasing the amount of carbon dioxide that gets inside the red blood cells. Inside the red blood cell, the enzyme carbonic anhydrase catalyzes a reaction with carbon dioxide and water, which forms carbonic acid. Carbonic acid then splits into a bicarbonate ion and a hydrogen ion. As more and more hydrogen ions start to pile up, the pH starts to fall. Now, both carbon dioxide and hydrogen ion can bind to hemoglobin. Carbon dioxide binds to the terminal amino acids in the globin subunits, forming carb-amino-hemoglobin, while hydrogen ions bind to the amino acid side chains that make up the globin subunits. And even though carbon dioxide and hydrogen ions don't compete for the same binding sites as oxygen and hemoglobin, they do stabilize the T state of hemoglobin. This results in a decrease in hemoglobin's affinity for oxygen, which forces the unloading of oxygen. So, for example, when muscles are working hard, like during exercise, they're producing a lot more carbon dioxide, and as a result, more hydrogen ions, which causes the pH to drop. So oxygen is unloaded in the tissues that need it most. Another factor is 2,3-diphosphoglyceric acid, or 2,3-DPG, which is a metabolic byproduct of glycolysis in red blood cells. When there are hypoxic conditions, like a lack of oxygen, which might develop when a person is at high altitudes, 2,3-DPG production increases. In the middle of the hemoglobin, there's a positively charged pocket between the four globin chains where the negatively charged 2,3-DPG is able to bind. Again, even though the 2,3-DPG doesn't bind in the same place as oxygen, it does stabilize the T state of hemoglobin, which decreases hemoglobin's affinity for oxygen, forcing the unloading of oxygen in tissues that are in need of it. Temperature also affects the strength of the bond between oxygen and hemoglobin. The heme component of hemoglobin, which is where the oxygen binds, contains iron, which helps anchor oxygen to hemoglobin. Higher temperatures affect this bond between iron and oxygen, favoring the T state of hemoglobin. So, using exercise as an example again, increased activity causes muscles to generate increased heat. And the rise in even a few degrees in temperature allows for more unloading of oxygen, and that helps meet the increased metabolic demands of the muscles. Finally, hemoglobin's affinity for oxygen can depend on the type of hemoglobin. For example, hemoglobin A, which is the main adult hemoglobin, has a lower affinity for oxygen than fetal hemoglobin. So, for example, this is important during gestation. Since a baby needs to compete for oxygen with mom, it's helpful to have a higher affinity for oxygen than mom to help efficiently transfer oxygen to the baby. So, in an adult with hemoglobin A, when partial pressure of carbon dioxide, temperature, or 2,3-DPG are high, or pH is low, hemoglobin affinity for oxygen decreases and more oxygen is unloaded in the tissues. The curve shifts to the right because now a higher partial pressure of oxygen, or P50, is needed to overcome these factors and saturate hemoglobin. On the flip side, when partial pressure of carbon dioxide, temperature, or 2,3-DPG are low, or pH is high, or we're looking at fetal hemoglobin instead of hemoglobin A, hemoglobin affinity increases, and less oxygen is unloaded in the tissues. The curve shifts to the left because a lower partial pressure of oxygen is needed to saturate hemoglobin. So far, to be clear, these factors affect hemoglobin's affinity for oxygen, but not hemoglobin's capacity to bind oxygen. This means that hemoglobin is still only able to bind 4 molecules of oxygen, but how easily it does so when there's a certain partial pressure of oxygen depends on the circumstances. Now, one additional situation to consider is when there's something like carbon monoxide floating around, which actually competes with oxygen for binding sites on hemoglobin. Hemoglobin's affinity for carbon monoxide is 250 times greater than that of oxygen. So hemoglobin will bind carbon monoxide, forming carboxyhemoglobin, more readily than it'll bind to oxygen. And once carbon monoxide binds, it doesn't unload easily, making those binding sites unavailable to oxygen. This has 2 effects. First, the affinity for hemoglobin to bind oxygen increases at the sites not filled by carbon monoxide. This is an attempt for oxygen to compete with carbon monoxide to bind to hemoglobin, but results in less unloading of oxygen in the tissues because of increased affinity. Second, the entire shape of the curve changes from sigmoidal to one that flattens out to a 0 slope. Where the curve flattens depends on how saturated hemoglobin is with carbon monoxide. For example, if 50% of the binding sites on hemoglobin are filled with carbon monoxide, the curve will flatten out at about 50% hemoglobin saturation with oxygen. And as the partial pressure of carbon monoxide increases, and more carbon monoxide binds, the curve will flatten out at lower oxygen saturation values. Alright, as a quick recap. The oxygen hemoglobin dissociation curve graphs the relationship between hemoglobin saturation with oxygen and the partial pressure of oxygen in the blood. The sigmoidal curve to the graph is the result of positive cooperativity, which increases hemoglobin's affinity for oxygen as more oxygen molecules bind. But factors like partial pressure of carbon dioxide, pH, temperature, 2,3-DPG, hemoglobin type, and carbon monoxide can all affect the affinity, causing a shift in the oxygen-hemoglobin dissociation curve and making hemoglobin more or less likely to unload oxygen in the tissues.